In this video, I will go over the whole of biological psychology. This includes the central nervous system, the effect of recreational drugs in the central nervous system, the structure of the brain and aggression, hormones and aggression, evolution and natural selection and aggression, psychodynamic approach, opposing the biological theory to aggression, Raine et al., Brendgen et al., the key question, practical investigations, correlations, twin studies, and scans. The CNS, also known as the central nervous system. Now, the CNS is made up of the brain and spinal cord. The brain communicates to the rest of the body through the spinal cord and allows messages to be passed from the body to the brain, as well as from the brain to other parts of the body, which leads to a response. The brain processes information from the senses and then is responsible for controlling the body's behavior that may result from this information. Cells in the central nervous system are neurons. A neuron is a nerve cell that processes and transmits information through electrical and chemical signals in the nervous system. This is a picture of a neuron. I don't think you're going to get asked how to label a neuron. However, I think you may get asked the function of a dendrite or nucleus or cell body. So I will tell you what their functions are now. So dendrites are at the far end of the neuron. They receive messages from other neurons in order to trigger an action potential within the cell. Carries electrical impulses from previous neurons towards the cell, and it's connected to other dendrites. Next you have the axon. The axon is the long bit of the neuron. It's a long branch from the cell body that passes electrical impulses down the end of the neuron to allow it to communicate with others. If you were to label it, it's that long yellow strip there. Next you've got the terminal buttons. They connect vesicles that store neurotransmitters. The axon terminal receives a message from other neurons, and this is at the far end opposite the dendrites. It receives messages from other neurons in order to trigger an action potential within the cell. It carries electrical impulses from previous neurons towards the cell. The cell body holds genetic information. It's where the part of the nucleus sits and contains cell nucleus, which houses genetic material for that particular neuron. It contains other material which allows the cell to function, for example, mitochondria for energy. The axon hillock attaches the axon to the cell body. The myelin sheath is a fatty layer that protects the axon and increases the speed of transmission. The nodes around there are gaps between the myelin sheath, which increase transmission as it is forced to jump between the gaps. I like to think of it as bunny hops between the gaps of the myelin sheath, and therefore that's why if you have increased layers of myelin sheath, it increases the speed of action potential because it forces the jumps more. Finally, Schwann cells. They surround the neurons and secrete myelin sheath. You also have to know the process of synaptic transmission. Synaptic transmission is when messages are passed through the body through neurons. Shown above is a picture of it. It's good to know the labels of this. You have the presynaptic neuron, that's pre the synapse. You have the postsynaptic neuron, that's after the synapse. You have the synaptic cleft, that's in the middle of the synapse. And you have the vesicles, they are filled with neurotransmitters. And you have the vesicle button, and that is at the end of the neuron. And you have receptors on the postsynaptic neuron that collects the neurotransmitters which are released from the vesicles. It's good to know how to label this, because when in your exam we need to explain this, you can draw it, label it, annotate it, and then explain it. So you need to know the process of synaptic transmission. This normally comes up as a 3 marker. 1. An action potential gets sent through the action hillock towards the terminal buttons where vesicles are located which are filled with neurotransmitters. 2. Once a message reaches the action calcium channels and opens and releases calcium ions. 3. Calcium ions triggers the release of vesicles. The vesicles will fuse with the outer membrane of the terminal button, releasing the neurotransmitters into the synaptic cleft. Receptors on the postsynaptic neuron will then absorb the appropriate amount of neurotransmitters through the lock and key method. Any neurotransmitters that are not absorbed by the postsynaptic cleft will be destroyed by enzymes in the synaptic cleft or will be absorbed by the presynaptic neuron by the uptake. Absorbed molecules will be destroyed by enzymes to turn off the neurons in preparation for future action potentials. Once the message reaches the axon, calcium channels open and release calcium ions. Now that is a lot to remember, and it was a lot of information, so here's the important things. An action potential reaches the synapse. That leads to the release of calcium. The release of calcium leads to the vesicles being opened on the presynaptic neuron, which leads to them going across the synaptic cleft, which leads to them getting absorbed by the receptors on the postsynaptic neuron. That was a very quick and shortened way to explain how you could shorten this down. However, please do shorten this down, as this is a very in-depth explanation of the process of synaptic transmission, and you already have a lot to remember. Action potential is a nerve impulse that passes down the neuron to stimulate the release of neurotransmitters, which is triggered by a change in electrical potential. A neuron is at rest at minus 70 volts, which leads to a cell being negatively charged. When a neuron is activated by a stimulus inside the cell, it becomes positively charged, which is at minus 55 volts. Negatively charged means there's less potential for an action potential, which is an inhibitory state. A positively charged means that there's more chance for an action potential, which is an excitatory state. Action of neurotransmitters. Dopamine has been related to emotion and cognitive function, as well as posture and control of movement. It has also been associated with the reinforcement in learning, as well as the dependency in addictions. Dopamine is used in hormone regulation, such as controlling the menstrual cycle. Serotonin is associated with mood control, particularly in the limbic system. It is involved with many other functions, such as feeling pain, sleep, and regulating body temperature and hunger. Noradrenaline is associated with emotion, particularly mood control. Noradrenaline is involved with sleeping, dreaming, and learning. Acetylcholine stimulates muscle contractions and has key function in motor control and movement. It's also necessary for memory and other cognitive functions such as attention and wakefulness and alertness. Acetylcholine is also involved in expressing some emotions such as anger and sexuality. So you need to know how recreational drugs affect the central nervous system. This typically comes up as a short answer question, however I have seen it as an 8 marker. So recreational drugs are drugs taken with no medical basis and are taken for enjoyment. They also bring function, for example, mood, perception and consciousness. For examples of recreational drugs are cocaine or heroin. Brain contains a reward pathway which causes you to experience pleasant or rewarding feelings. This encourages to repeat a behavior that activates the pathway. However, recreational drugs hijack this reward system and produces the pleasurable feeling without any adaptive functioning. An example of a reward pathway is exercising and then feeling good because you've exercised. You get the reward feeling for doing something good. The mode of action describes how a drug or any other substance produces an effect in the body. Drugs change the way neurotransmitters work. Most drugs have an impact on the dopamine system. Specific examples of drugs and how they affect the central nervous system. Opioids, for example heroin or morphine, they reduce GABA activity. This leads to an overactivity of dopamine neurotransmitter in the reward pathway of the brain. Cocaine, this increases the activity of dopamine pathway by blocking the uptake of dopamine. Alcohol, this has a depressive effect on the nervous system. It acts to inhibit the neurotransmission by increasing the action of GABA. If you don't know what GABA is an inhibiting neurotransmitter. Nicotine targets aspects of the dopamine pathway, increasing the amount and transmission of dopamine by blocking enzymes that break it down. This mimics acetylcholine and binds to the nicotine receptors. Cannabis binds to cannabis receptors in many regions of the brains. Dopamine neurons do not have these receptors, so instead cannabis blocks GABAergic neurons. Amphetamines, they work by increasing dopamine and noradrenaline in the synapse, by changing the reuptake process so it works in reverse. Amphetamines force the release of these neurotransmitters and can block reuptake. In high doses, it can inhibit their breakdown by enzymes. Individual differences. How drugs and their effects change to person despite sharing a common biological mechanism. Expectations. A mood or what they expect from a drug can affect how the dopamine system responds. If someone believes that a drug will make them feel good, the brain's reward system may make the experience feel stronger. Younger people have a developing dopamine system, making them more sensitive to drug rewards, whereas older people often have fewer dopamine receptors, so they may experience weaker drug effects or need a smaller dose. Fat-soluble drugs are stored in fat cells, which may cause them to have a longer-lasting effect in people with more body fat. Fat cells don't have as much dopamine receptors. People with more muscle may metabolize drugs faster. Liver and kidney function. If a person's liver or kidney doesn't work well, then drugs may stay in the body for longer, overstimulating the dopamine system and increasing the effect. Conditions like ADHD or depression that involve changes in dopamine. People with these conditions may experience drugs differently, such as stronger or weaker effects, depending on their dopamine system and how it works. Regular drug use can reduce the number of dopamine receptors, meaning people need more of the drug to have the same effect. Withdrawal. After stopping the drug, the brain may become sensitive to dopamine, making people crave the drug more or feel the stronger effect if they use it again. Strengths. The supporting research is lab-based, and therefore they have controlled procedures which are standardized. They are credible in saying that the reward pathway relates to dopamine, which is a neurotransmitter giving pleasure, and recreational. Drugs work on the reward pathway to increase pleasure. The credibility comes from observation of those who take recreational drugs. They tend to experience extreme highs, addiction, and withdrawal. There's a lot of supporting evidence. Olds and Miner, 1954, found that pleasure center in the brain in rats. Stryker et al. 2012 looked at the effect on cannabis and found an effect in the hippocampus of mice. However, this theory is reductionist. There's complexity in how the transmission works in the brain that is hard to capture. Current tools cannot measure transmission. Cannabis limits hippocampus activity, but also leads to more dopamine activity, and there's more than one reward pathway. Having one explanation for the mode of action of recreational drugs seems rather simplistic. Conducting human research is difficult. Scanning and finding out about receptor activity is not straightforward. Scanning needs to be more sophisticated, and the focus is currently on the transmitters that are easily studied. Therefore, all conclusions are about transmitters. There may be others which we cannot study in this moment in time. The use of animal studies as supporting research is hard to extrapolate to humans. It's difficult to study transmission. Current tools cannot measure transmission. For example, cannabis research focuses on the side effect of GABA and glutamate because these are easy to study. Having one explanation for the mode of action of recreational drug is simplistic. Now, there's many different types of aggression, for example, verbal, physical, indirect. However, you need to know about four main types. Person-oriented aggression is where the goal is to cause harm to another person. This can be through physical or verbal aggression. It occurs through showing hostility to another person. Instrumental aggression is where the goal is to obtain some kind of reward or preferred outcome. This can be played out through the coercion of another person. Reactive, a response to a hostile or aggressive situation. Aggression is a shown response to provocation. Sanctioned aggression. Aggression is behaviour which is either legal or permitted because the ultimate outcome is justifiable. Brain structure. Now I will be going over the lobes of the brain change over time, the limbic system, the right and the left hemisphere, as well as more key features. The reason I will be doing this is I have seen questions on paper 3 about the left and the right hemisphere, which is GCSE knowledge. However, it came up. Even though this may be in more detail than needed, it may come up on your paper 3 paper. Some key words you may need to know. Tree planning is to drill holes into the skull. Phenomenology, mapping bumps on a person's skull and using this to deduce aspects of their character. Lateralization. The brain has two hemispheres which differentiate. There's evidence in fossil that tree planning was used in connection to migraines and epilepsy, suggesting that over 10,000 years ago, humans had some idea about brain functioning. Hippocrates, a Greek physician born in 460 BC, put forward the idea that each side of the hemisphere had different function. This is the idea of lateralization. 19th century phenotology was used by Franz Joseph Gall. The case of Finnish gates provided more insight in 1849. Broca found the area of the lower left lobe, Broca's area, responsible for motor control, speech production, and if the Broca's area was damaged, the individual could not understand, but they could speak. This is called Broca aphresia. So you definitely need to know about the different lobes of the brain, and you also need to know what is in the different lobes of the brain. For example, the prefrontal cortex is in the frontal lobe. The frontal lobe is located in the front of the brain. The main functions are associated with higher cognitive function, including problem-solving, decision-making, attention, intelligence. The frontal lobe contains the motor cortex, which is responsible for planning and coordination. It also contains the prefrontal cortex, which is responsible for attaining a higher cognitive function, and the Broca's area, which is responsible for language production. Prefrontal cortex helps with attention, emotion, self-control, and decision-making. The prefrontal cortex takes time to fully mature and doesn't finish fully developing until your mid-20s. Damage to it causes conditions like ADHD or depression. You can strengthen it with mindfulness, learning, and staying active. The temporal lobe is located on both sides of the brain, and its function is to understand language, memory, auditory information processing. The parental lobe. This is located at the top of the brain between the frontal and occipital lobe, above the temporal lobe. The parental lobe is essential for integrating information from the body's senses to allow us to build coherent a picture of the world around us. Stoma to sensory cortex. This is receiving and processing sense information. The parental lobe allows us to perceive our bodies through stoma information to sensory information. It helps us with visual spatial processing, reading, and number representations. The occipital lobe is located at the back of the brain behind the temporal lobe and the parental lobe, and below the occipital bone. It receives sensory information from the visual data. Some functions include being able to access size, depth, distance, determine colour of information, object, and facial recognition, as well as mapping the visual world. It contains the primary cortex, which receives sensory information. The limbic system is a complex network of interconnected brain structures that plays a vital role in regulating human behavior, emotion, especially those tied to survival instincts. It contributes significantly to memory processing and motivation. Parts of the limbic system are the thalamus, the pineal gland, the hippocampus, the cerebellum, the amygdala, the pituitary gland, the hypothalamus, the cerebellum, and the basal ganglia. Thalamus. It relays information between the cerebral cortex and the brainstem. It has an interactive role in relaying sensory information and motor information. It contributes to many purposes, for example, attention, preparation, and timing. The pineal gland regulates the sleep-wake cycle and circadian rhythms by producing melatonin in response to light and dark signals. Melatonin also impacts moods. The hippocampus is associated with learning and memory. It plays a key role in forming memories, serves as an early storage system for long-term memories, and helps to convert memories into permanent memories. The cerebellum plays a key role in physical coordination as well as regulating emotions and thoughts. It's located in the back of the brain behind the brainstem and aids in motor skills. The amygdala is involved in processing emotions and fear. It ties emotions to meaning, processing of rewards, and helps us to make decisions. It's linked to fight or flight. It's associated with emotions and aggression. The left side of the amygdala links to the reward pathway and positive and negative emotions. The right side of the amygdala links to negative emotions and sadness. The pituitary gland is also known as the master gland. It releases hormones into the bloodstreams and forms a crucial link between emotion processing, survival behaviors, and hormone regulation. Hypothalamus modulates range and physiological functions, controlling autonomic functions, for example, hunger. It integrates information from parts of the brain and responds to various stimuli. The hypothalamus is connected to the pituitary gland and is responsible for stimulating or controlling the release of hormones from the pituitary gland. Cerebellum monitors and regulates behaviors. It helps posture and balance. The basal ganglia is a group of structures that regulate the coordination of fine motor movements, balance, and posture alongside the cerebellum. The basal ganglia is connected to motor areas and links the thalamus with the motor cortex. It is involved in cognitive emotions and behavior. The cerebellum is the largest part of the brain. It consists of grey matter and white matter. It contains the lobes of the brain, the frontal lobe, the parental lobe, and the occipital lobe. It produces a high-functioning role, and it is also divided into the right and left hemispheres. The left hemisphere controls the right-hand side of the body and receives information from the right visual field, controlling speech, language, recognition of words, letters, and numbers. The right hemisphere controls the left-hand side of the body and receives information from the left visual field. It controls creativity, context, recognition of objects, places, and faces. The left and the right hemisphere is connected by the corpus callosum, which allows the two hemispheres to communicate and allows information being processed on one side of the brain be shared with the other side of the brain. Finally, ventricles produce cerebral biospinal fluid, which helps cushion the brain from any blows to the head and provide nutrients. High pressure leads to blockage in ventricles, which can cause hydrocephalothalamus. The hypothalamus regulates eating, drinking, and motivated behaviors. The hippocampus, the short-term memory passed to the long-term memory through the hippocampus. The cerebellum coordinates movement and balance as well as stores memory and learning skills. It's vital in balance and coordination. The medial temporal lobe, you can shorten this down to MTL. This plays a large role in memory, emotion processing, and it's where the hippocampus is located. The striatum plays a role in motivation. Lastly, the thalamus acts as a brain relay station, processing and directing sensory and motor signals to the correct areas of the cerebral cortex. People with schizophrenia were found to have a significantly reduced volume compared to those without schizophrenia. The midbrain contains an area called the pre-anterior grey matter, or PAG, which links the amygdala to the hypothalamus with the prefrontal cortex. It has a role in coordinating and integrating behavioral responses to perceived internal and external stresses, such as pain and threat. Supporting research is Lowenstein and Stern, 1988. Lesions to PAG in rats were shown to increase aggression when rats were confronted with potential threats in the form of unfamiliar male rats being introduced to the cage. The amygdala is the center for emotions, emotional behavior, and motivation. It integrates internal and external stimuli, and every sensory modality has an input, which when combined gives us an instinctive feeling or reaction to the environment, which will include aggression. The prefrontal cortex also connects to the amygdala. This connection may lead to aggression. Supporting research, Brendgen and Luttrell, 2005. They carried out a meta-analysis where they analysed results in 15 brain studies on aggression. They found all brain scans had a less active prefrontal cortex and amygdala, which led to aggression and aggressive behaviours. Thalamus. The role of the hypothalamus is to maintain homeostasis through the regulation of hormones, including the regulation of sexual functions. This is linked to aggressive behaviour in males via the production of testosterone. Supporting research, Egger and Flynn, 1963. Egger and Flynn found that electrically stimulating parts of the amygdala would either increase or suppress aggression. In their study, they also demonstrated this by stimulating areas of the hypothalamus, causing cats to show aggressive behaviour towards rats. This suggests the hypothalamus has a role in aggression. Prefrontal cortex is an area in the brain that sits right behind the forehead. It influences social interactions and regulation of behaviour. Prefrontal cortex damage has connections to the amygdala and hypothalamus. Damage to this area often leads to a problem with anger management, irritability, and impulse control. Supporting research, Raine. He wanted to find out the difference in brain structure between NGRIs and non-murderers. He found that there was less activity in the frontal lobe, especially in the prefrontal cortex. Limbic system. The limbic system includes the hippocampus, the amygdala, and the hypothalamus. It has a role in self-preservation and fight-or-flight response. It's responsible for controlling emotions and for focusing attention on events that are emotional for the individual and links the thalamus and the hippocampus. Supporting research is Andy and Valdemar Marty, 1987, which stimulated parts of the hypothalamus and basal ganglia, parts of the limbic system of cats, and had aggressive seizures. Phineas Gage's brain damage led to his understanding that a damaged prefrontal cortex could increase aggression. Studies in rodents like cats have found that different areas of the brains are associated with different things. Studies in rodents of cats have found that different areas of the brains are associated with different types of aggression. Offensive behaviours is associated with the middle of the hypothalamus. Defensive behaviour is associated with the dorsal back of the hypothalamus, and predatory behaviour is associated with this side of the hypothalamus. Issue of social control. The application of these findings can be used to treat aggressive patients by performing lobotomies to the prefrontal cortex, which subdue emotions and emotional reactions. As well as this, we can start looking up people who have an overactive amygdala before they've committed a crime. This proves to say that we do not know the cause and effect relationship. We do not know if the crime is caused by brain structure or if brain structure is there in the first place. Animal evidence cannot be generalized to humans, as this is reductionist. Also, humans are logically driven by their actions. It's not just instinct. We can think about things like morality. Animals can't. However, animals do share basic functioning. Mice share 70% of the same brain structure to humans. Scanning is reliable, but validity is questionable, as this measures processing of an artificial task which are not like real life. Ethical issues of using animals and performing lobotomies counterbalance with the other explanations like evolutionary approach. And this only considers nature, not nurture. For example, family background needing money, or the psychodynamic approach. There's a lot of supporting evidence from case studies and animal studies, as you've seen. We now have more accurate scanning techniques like PET scans and fMRIs. We use standardized scientific procedures to make it a reliable way to diagnose and test aggression. Genetic research supports these findings. The endocrine system. The endocrine system secretes hormones from glands which travel in the bloodstream. Using hormones, the response is slower than the nervous system. It produces a longer-lasting response due to using hormones instead of neurons. The endocrine system's main function is keeping homeostasis, which is keeping our bodies at a constant. Our hormones levels are regulated by feedback signals sent from the hypothalamus to the pituitary gland, which causes the pituitary gland to secrete a stimulating hormone into the bloodstream. The levels of this hormone increase in the bloodstream, the hypothalamus stops secreting hormone and the pituitary gland shuts down, and the secreting of the stimulating hormone shuts down. This is feedback. How does feedback loops work? The hypothalamus sends a hormone to the pituitary gland, and the pituitary gland secretes a stimulating hormone. The target gland releases its hormone, leads to the level of the target gland's hormone in the bloodstream increasing. The hypothalamus then stops secreting the hormone to the pituitary gland, which leads to the pituitary gland stops secreting the stimulating hormone. The target gland stops releasing its hormones. This is because everything should be at optimum function. Therefore, the hormones are no longer needed. Sympathetic and sympathetic nervous system. The endocrine system works alongside the nervous system and automatic nervous system with fight or flight response. The parasympathetic and sympathetic nervous system are integral parts of the fight or flight response. When danger has been identified, the hypothalamus triggers the responses below. This is because the sympathetic nervous system will stop unnecessary processes. Once your body realizes it can relax, the parasympathetic nervous system kicks in, and everything to normal. Hormones and aggression. Cortisol. Cortisol is produced in the adrenal glands, and its role is to manage stress, and it tends to inhibit aggression. People with lower levels of cortisol tend to be more aggressive because their autonomic nervous system is unaroused, and cortisol is not inhibiting aggressive behavior through fair response, so people are more likely to act impulsively. On this slide, the support for high and low levels is mixed up. So research support for low levels is Vercom, 1985, which reported low levels of cortisols in violent offenders, and Tensi and Kyrie, 1985, reported low levels of cortisol in aggressive school children. Bookhaven, however, 2005, found reactive aggression was associated with high levels of cortisol. Testosterone. Testosterone is a chemical that maintains male characteristics. It's higher in males than females. Exposure to testosterone can lead to increased spatial ability and competitiveness. As an adult, it can affect aggression. Support for high testosterone. Wagner et al., 1979, castrated mice and observed that aggression levels dropped. When castrated mice are injected with high testosterone, their aggression levels rose to pre-castrated levels. Dabbs et al., 1978, measured the level of testosterone in violent and non-violent crimes and found higher levels of testosterone in violent crimes. Mazza and Booth, 1998, found that men with high levels of testosterone are more likely to be in a divorce, single, be arrested for offences, buy or sell stolen property, be in debt, or use weapons in a fight. Serotonin and dopamine. Low levels of serotonin are associated with increased aggression because it inhibits aggression due to the depressive effect. There are links between dopamine and aggression as well. Dopamine is a neurotransmitter linked to attention and pleasure. Increased levels of dopamine are associated with aggression, and antipsychotic drugs seem to reduce aggression and aggressive behaviors. et al. 2003 measured the level of serotonin and dopamine in rats when they were expecting a fight, and they found the level of serotonin and dopamine increased when they were expecting a fight. Strengths. There's a lot of supporting research, which we've gone over. Brain imaging techniques have been proven to be reliable to show a correlation between brain activity and aggression. Studies have used careful controls, which give it scientific credibility. Constant findings have linked cortisol to aggression. Both humans and animal studies link testosterone and cortisol to aggression. There's an overwhelming amount of research support for both human, children, animals, and adults linking testosterone and cortisol to aggression, suggesting reliability. Our research support uses animals to prove hormones lead to aggression. This can't be generalized as you cannot extrapolate animal behavior to human behavior, therefore it cannot be applied. Other explanations. Nature, not nurture. It only explains how your nature can cause aggression and not how your nurture can cause aggression. This is a reductionist explanation of aggression as it only focuses on one side of the argument. Brain scanning techniques lack validity as they aren't doing natural tests in their natural environment. As well as this, correlation between scans doesn't actually prove that behavior is caused by brain activity. Tanner et al. 2011 showed that the complexity of human behavior means that researchers must be willing to look beyond purely hormonal explanations and draw on other variables to have a holistic view. Evolution and natural selection and aggression. Key definitions before we start. Genes are the fundamental unit of heredity that contains DNA that will be passed on to the offspring. Mutation, a change in genetic structure in an organism that makes it different from other same kinds of organisms. Evolution, change in heredity characteristics in biological populations over successive generations. Natural selection, the gradual process in which inheritable traits become more or less common to adapt to environment. Sexual selection, individuals that succeed in attracting a mate outproduce others in the population. What is evolutionary theory? The structure and function of the brain has changed over time. Successful humans were those best suited for the environment and reproduced. Psychologists at the EEA use fossils to understand and speculate what type of behavior is adaptable, to try to determine what behavior is genetically determined through brain structure and chemistry. However, our environment has gone through rapid change and therefore we may not need some behaviors that may have helped us in the past. For example, aggression. Explaining aggression between males. The evolutionary approach to aggression sees aggression as an ability to increase survival chances and therefore increase reproductive success. Example of behaviors that have come about due to evolution. Deterring males from infidelity. Aggression towards the other sex so they stay as a mate. Deterring rivals from future aggression. Aggression to dominate. Negotiating status and power and hierarchies. Aggression to gain dominance, money, same-sex members to aid selection. Gaining territory, E.G. warfare. Inflicting costs on same-sex rivals. Aggression between same-sex for resources/mates. Defending against attacks. Aggression to prevent loss of resources, status, e.g. bullying to be popular in school. Aggression between females. Females should be less physically aggression. They have an evolutionary disadvantage due to their nature. Females in nature tend to be less aggressive due to having to look after children, due to having to breastfeed, due to their having a nurturing side to them, due to having to have this, due to having kids. To put themselves at risk is to having to put their family at risk. This leads to women having more verbal and emotional aggression than having physical aggression. When competing for mates, women use verbal aggression. This is shown by Buss, 1999. Women had to compete for mates and put other women down to make them seem less attractive when trying to reproduce. Jealousy. Jealousy is an emotional response to an anticipated loss of an affection or status. Males need to compete with other males to access a choosy female. Lower status men are willing to take genetic risk by using aggression or they will face extinction. Infidelity is unfaithfulness of sexual partners. There are two types: emotional infidelity which is a form of affection or attachment to another person, like flirting, and sexual infidelity, which is any behaviour involving sexual contact. If a female is unfaithful, the male faces potential paternity uncertainty. This can lead to male risking wasting an investment on an offspring which is not their own. This can lead to jealousy, violent aggressive jealousy relating to female infidelity. However, infidelity is less harmful to females, as females can always guarantee that the offspring is their own. Females do have to compete, however, for quality of men. Therefore, women take fewer risks with violence and use indirect forms of aggression. 40% of domestic violence victims are men, 2009. However, if a man is unfaithful, females risk losing his resources, energy, protection, and commitment to the children. Strengths. Daly and Wilson, 1988. Humans' violent aggression and homicides are more common in males. Daly and Wilson, 1988. Applied to social living, deterring females from infidelity. Young wives/girlfriends are more likely to be killed than older ones. This is reliable, however outdated. This theory explains gender differences. It explains why males are more likely to be aggressive than females. There's evidence from Manson and Raine, 1991, who found that female chimps who were cooperative were selected via males, and male chimps who were aggressive were selected by females. However, the research do not have cause and effect, and variables were not measured nor manipulative, and you're comparing chimps to humans. The application to reproductive behaviour, we choose partners who will enhance our reproductive success. David Buss, 1989. Across 33 countries, males preferred younger, more attractive women who were easier to reproduce with, and females preferred older, healthier, and ambitious, hard-working men. As well as this, Darwin's theory of natural selection is well established and has supporting evidence towards it. Weaknesses. Nurture. Adopting and fostering. Males can care for a kid that isn't theirs. This could be due to personality. This theory does not look at how nurture may affect someone. Therefore, this is not applicable. Differences between culture and aggression. Wolfgang and Verekutai, 1961. Amongst the Krongsang people of Kalahari, aggression was discouraged from childhood, whereas in Brazil, aggression is accepted. Therefore, this study is not applicable to everyone. Researcher bias. The EEA based the theory of fossils. Therefore, this could be biased, as we do not know what fossils actually tell us. However, Lande Vias found that male fossils were significantly bigger than female fossils. The size difference demonstrates that the existence of male-on-male competition. It's impossible to test that evolution actually happened, and other theories such as the psychodynamic, hormones, and brain structure can be used to counterbalance this theory. The psychodynamic approach to aggression. This opposes the biological theory to aggression. So we have an id, an ego, and a superego. Our ego is the conscious, the superego are preconscious, the id, the unconscious. The ego, the conscious, is 2 years plus, and it controls and delays the urges of the id. The development of our norms and values are learned in our ego. It understands when and to the extent of which a behaviour is appropriate. The ego has no problem with using aggression as long as it thinks it will win. For example, you may use social aggression in the ego to make someone feel bad about themselves, but you will win. You will make them feel bad about themselves. They will not hurt you back. Superego, or preconscious. This develops at 3 to 6 years old. Morality principle develops. Pride when behaving correctly, guilt when behaving incorrectly. Aggressive behaviour or impulses are controlled by the superego. The id or unconscious. You are born with this. This is a primitive part of personality. It is where Eros and Thanos resides. It's entirely impulse, with no thought for consequences. If the id is denied pleasure, it becomes frustrated, which leads to aggression. If any issues in developing ego or superego, it leads to you not being able to control the I did, which leads to aggressive behaviour or urges. Eros and Thanos. Eros is focused on preservation and enjoyment of life, whereas Thanos is driven towards death, destruction, and initially this is directed at the self, and then it's redirected towards others in the form of aggression. Eros and Thanos balance each other. Defense mechanisms to prevent aggression from being directed at oneself. Thanos. Repression. Aggressive urges that we repress drain libido. We don't have enough energy to keep aggression repressed. Therefore, it explodes out. Denial. The conscious mind refuses to admit what they are doing. Projection. The superego hostility is directed towards people who seem to be acting on the same feeling, often involves victim blaming for provoking aggression. Sublimation. I did urges are acted upon but transformed into something socially acceptable. Aggression may be sublimated into sports. Displacement. ID urges are acted upon but they are directed at different targets. This often happens with aggression, which can be targeted at someone or something else, rather than the real cause of your anger, e.g. sports. Now, sublimation and displacement sound very similar, and they are. However, with sublimation, it's socially acceptable aggression. With displacement, it doesn't have to be. You could displace your anger onto your boss, onto your friend, onto your family, and it could not be socially acceptable. It could just be anger. But you're not actually angry at them, you're just displacing this anger. Psychodynamic theory can explain different types of aggression. Social aggression comes from defense mechanisms because the superego won't allow us to act out physically our aggression, and to appease the superego, the ego acts out social aggression. Most social aggression is displacement, anger at parents, aggression towards peers, or sublimation. Sexual jealousy. Sexual feelings come from the I did but are controlled by the superego. We learn to sexually shame from our parents and wider society, but its sexual needs are insistent. This leads to conflict and unhappiness. Repression enables us to act normally but drains the eros, and displacement of frustration leads to us taking this frustration out on others. Strengths. Catharsis is an ordinary experience. Many people do feel calmer or more at ease after venting aggression through sport, gym, or games. This means the theory is applicable to real life and reliable. Psychoanalysts can treat aggression. Psychoanalysts help clients by exploring dreams and childhood memories to work out what they mean, and application is allowed, however, not measurable. Freud based his theory on psychoanalysts and his patients. He also carried out a case study on little hands. As well as this, he psychoanalyzed himself, studying his own dreams and childhood memories. This is scientific and reliable. A strength and a weakness is that his study is non-falsifiable. It cannot be proved, nor can it be unproved. This is because there is no reliable evidence that the I did, eros, and superego actually really exist in our brain. Weaknesses of the theory then. It doesn't explain aggression entirely. Couples who have an argument should be less likely to be aggressive due to letting out aggression. However, couples who argue are more likely to be abusive. Therefore, the theory is not applicable or valid. Other explanations for aggression. For example, the brain. This theory reductionist as it doesn't see the biological approach. Karl Popper. True scientific theories are falsifiable. Popper states that theories cannot be disproved not because of lack of evidence, but because they are vague. This shows that Freud's theory is not scientific. Raine et al. 1997. Brain abnormalities in murderers, indicated by PET scans. NGRI means not guilty for reason of insanity. Scans, also known as position emission tomography, injects a person with a small amount of radioactive material to conduct a scan. The radioactive substance is attached to the glucose. A task is given to patients to do, whilst the glucose gets used up and the radioactive substance breaks down and emits positions. Where there's a high concentration of gamma rays, there's a high concentration of activity. This is where glucose will be used up. This is shown by warmer colours on the PET scan. You can see this on Raine's actual PET scans. Strengths of using PET scans is that I can help predict what issues patients may face in relation to brain activity. They also detect areas of damage indicated by which parts of the brain show abnormality. However, a weakness of using PET scans is that you're not advised to have too many, as it's unclear the future effects, and it's an invasive technique, and you may interpret them incorrectly. Or, if you know the results of the study, you may interpret them biasly. Previous research carried out by Raine showed that those who pled not guilty for reason of insanity had a significant difference in brain activity in the prefrontal cortex in comparison to controls. The study had an animal basis, as he studied areas of the brain which had proven to have high activity in animals. Aims. Raine wanted to investigate whether other areas of the brain associated with aggression would be different in murderers to control participants. He wanted to find a difference in brain structure activity between those who had committed murder and non-murderers. He wanted to investigate the role of the prefrontal cortex, corpus callosum, amygdala, medial temporal lobe, hippocampus, and thalamus in aggression. Now, Raine had 41 criminals, 2 females and 39 males, who were convicted of murder or manslaughter, awaiting tests to prove NGRI. They had a range of disorders from schizophrenia, substance abuse, epilepsy, learning disability, and paranoid personality disorder, all taken off medication 2 weeks prior to the scan. Now, Raine used a matched pair design, and due to this he had 41 control participants who were matched on age, gender, screened for general health, and had a psychoactive interview took place. Participants were excluded if they had a history of seizures, head trauma, or substance misuse for their own good. Procedure. Raine used a continuous performance task consisting of blurred numbers to focus on. They practiced 10 minutes before the injection of glucose tracer and completed the task for 32 minutes. Then the PET scan was taken. You can evaluate both the participants and the procedure. Due to only using 2 females and 39 males, it's low in generalizability to females' NGRIs. It also can be seen as reductionist due to ignoring factors. It's shown that the NGRIs had a range of disorders: schizophrenia, substance abuse, epilepsy, learning disability, paranoid personality disorder. All these things could have led to them becoming criminals. Raine used a matched pair designs. This should control for individual differences, therefore making it reliable. Due to screening control participants, it can be seen as highly controlled for extraneous variables. It can also be seen as ethical. Due to using a continuous performance task, it can be seen as low in ecological validity and mundane realism because this does not measure aggression. Results. NGRI showed less activity in the frontal lobe, especially in the prefrontal cortex, which is associated with rational thinking, self-resistance, and memory. Less activity in the parental lobe, associated with abstract thinking. More activity in the occupational lobe, associated with vision. Less activity in the corpus callosum, which is the bridge of nerve fibers connecting the 2 hemispheres of the brain. This can lead to someone being more impulsive. An imbalance between the left and the right hemispheres in the limbic system. Less activity on the left side of the amygdala, more on the right side of the amygdala. The medial temporal lobe and the hippocampus. More activity on the right side of the hypothalamus. These areas are associated with aggression in animals. Conclusion. There's less activity in the prefrontal cortex, which leads to self-resistant and memory issues. This means you're better at remembering consequences. Less activity in the parental lobe, which leads to no sense of good or bad. A limbic defect leads to aggression, which is observed in cats. This is supported by Andy and Vitelli, 1987. Less activity in the corpus callosum makes it harder for the brain to communicate. Strengths. Raine used a matched pairs design with a large sample of 41 criminals and 41 controls, all with the same gender, same age, same ethnicity. This makes it valid. The scans are reliable comparisons between groups, and they all had the same procedure. It allows for different brain activity to be compared objectively and reliably. Raine's study was based on animal studies, for example, Andy and Vitality, Egner and Flynn. This makes it reliable. It can be applied to society, and it could be done to identify criminals and to treat aggressive criminals, as well as to give people, like NGRIs, appropriate sentences. It's reliable. Bufkin and Luttrell, 2005, carried out a meta-analysis of 17 brain studies, and they found out having a less active prefrontal cortex and amygdala leads to aggression. Weaknesses. It's not representative. There's a small number of offenders and only 2 of the offenders were females. It's not a good explanation of violent offenders. Possible variation in procedural images are based on location of certain brain landmarks within each individual. This can lead to incorrect results. PET scans can be unclear and interpreted differently. Bias by professionals may lead to different interpretations. It's impossible to be sure that brain dysfunction is related to behavior. Extraneous variables can affect findings, or social or situational factors can lead to violent behavior. This makes the theory unreliable. Results cannot conclude whether violent behavior is biological or environmental. The extrapolation of animal to brain to human is reductionist. Animal brains are much smaller, and humans can think about complex processes such as morality. Raine even said this doesn't fully explain criminal behavior. Genetics is not the only reason why people commit crimes, and treatment is unjustifiable. Adoption and twin studies. What are twin and adoption studies? Twin and adoption studies are a way to investigate nature and nurture. In general, twin studies are to investigate the role of nature, genes, and adoption studies are ways to investigate the role of nurture, upbringing. Adoption studies and twin studies are always natural experiments. This is because the IV being investigated, whether you're biologically related to your family or what type of twin you are, is naturally occurring. You cannot manipulate variables like these. It would be morally wrong to force your child to be adopted and to see what happens to them. And it's scientifically impossible to cause twins to be born. However, fertility treatments can increase the likelihood of twins, like IVF. There are 2 types of twins, monozygotic twins and dizygotic twins, MZ and DZ. Monozygotic twins are twins conceived in a single egg, which will split later. These twins share the same genes. This means they must share the same sex. They are also called identical twins, and they usually look identical, although development in the womb and later health and diet can make them look different. Dizygotic twins are conceived when 2 or more eggs were fertilized at the same times. These twins share the same amount of genes as any brother or sister, up to 50%. These twins can be different genders. They are also known as fraternal twins, and they don't have to look identical, although they may be hard to tell apart. Over time, in the past, there was no 100% way of identifying MZ to DZ twins. There's a big grey area for similar-looking same-sex twins. However, 2 inventions since the 1970s have changed that. Ultrasound scanning may reveal which twins are sharing a placenta or the same membrane, which have massively increased the likelihood that they are MZ twins. Genetic testing can be carried out to reveal, with much greater certainty, which twins are MZ twins. Twin studies involve comparing MZ twins with DZ twins. MZ twins share 100% of their genes, and DZ twins only 50% of their genes. Studying the behaviours of twins reveal a lot about the influence of nature. If MZ twins and DZ twins behave the same way, it suggests that nature does not play a role in the role of behaviour, because the twins share the same environment, nurture, which encourages them to be the same. If MZ twins and DZ twins behave differently, it suggests that nature is at work because they share the same environment, is not producing the same behaviour, so the genetic differences would explain this. Twin studies are measured using concordance rates. Strengths of twin studies. Because of fertility treatments, more women are having twins. This makes it possible for us to study twins in large samples, making it more reliable. Twin studies can tell us whether important behaviours are inherited. For treating disorders like schizophrenia, it's important to know if this is inherited, to know if children are at risk. To an example of a naturally occurring variable means researchers cannot manipulate this variable themselves. This reduces the risk of researcher bias. Most psychologists argue that there's so much benefit from twin studies, so they have a social responsibility to study twins. Moreover, most twins are only subject to the sort of attention and curiosity they would only receive in day-to-day life. Weaknesses of twin studies. Twins are rare, 1.5% of the UK births, and MZ twins are rarer, 0.5% of UK births. Therefore, this is not a representative sample. Twins have unusual lives. They attract attention and often get treated differently. And they have different personalities. And they get mistaken for the other. Therefore, they're not generalizable to the general population. The identification for MZ and DZ twins still aren't perfect. For many studies like Gottman and Shields in 1966, they did blood tests and fingerprint combinations. However, they still didn't know if the MZ and DZ twins were actually MZ and DZ twins. 94% of the time, the allocation of MZ and DZ twins were right for Brendgen's study, and Brendgen considered this to be reliable enough. Confounding variables. Just because twins are brought up in the same environment doesn't mean that their upbringing was exactly the same. They have different lives and different life events. Biological determinism. The idea that your genetics entirely shape your person and who you grow up to be. In reality, we are influenced by a mixture of environmental and genetic factors. This limits the usefulness of twin studies because they don't show a concordant rate of 100. The best they can show is MZ being higher than DZ, suggesting that genetic has an influence. However, it doesn't entirely cause behavior. BPS Code of Conduct warns against damage of self-confidence. Twin studies may make twins feel weird, unusual, or different, and this is against the BPS Code of Conduct. Gottman and Shields, 1966, twin study. The aim was to investigate the relative importance of genetic and environmental influence on schizophrenia by comparing MZ and DZ twins. Participants. The sample was gained through secondary data from a hospital records of twins. 24 MZ pairings and 34 DZ pairings of the same gender with equal number of males and females aged between 19 to 64. Procedure. Case history providing a record of verbal behavior, a personality test, a test used to measure disorientated thinking, conduct on twins and parents. Concordance rates for schizophrenia in the twins of schizophrenics were used to analyze the results. Overall concordant rates 42% MZ, 17% DZ, twins with severe schizophrenia, 5% MZ and 22% DZ. Conclusion. There is a higher concordant rate in MZ twins, but it wasn't significant. It's concluded that both environment and genetic factors plays a role in the development of schizophrenia. Strengths and weaknesses. Generalizability. Twins are unique, sharing 100% or 50% of DNA. Most people don't have this, so it's unrepresentative. However, both men and women were used from ages 19 to 16. Reliability. Gottman based the study on 11 other studies and got similar results. Reliability. In the 60s, DSM-1 was used to diagnose schizophrenia. Application. It's useful for families of histories with schizophrenia, as it's useful to know whether it's genetic, so therefore they should avoid triggers, or if it's environmental. Validity. It assumes that any differences in concordance rates is due to genetic factors. However, it doesn't consider confounding variables like environments, which the twins both share. High in internal validity. Twins share similar environments as well as they were brought up in the same family, so results may be accurate. Ethics. They had informed consent from the twins. Ethics. Mentally ill patients will not understand the research or what consent is. Adoption studies look at the impact of nurture on children who are raised by parents who are not their biological parents. Because there is no biological connection between the parents, if the child grows up to share the parents' traits, the traits are probably produced by nurture. Adoption studies are made more valid if the researcher has information about the child's biological parents. If the child grows up with traits that resemble the adoptive parents more than the biological parents, then this is stronger evidence that the traits are down to nurture. Adoption studies are usually measured using correlational techniques. The researchers are looking for a correlation between the behavior of the child and the parent. Strengths. The child's anonymity is preserved and this respects their privacy. Adoption studies can tell us whether good parenting can correct bad genes. Social responsibility is important in ethical research. 5,330 children are adopted every year. Therefore, when we do research like this, we can understand how adopting and how nurture can affect us. Weaknesses. Adoption studies may have used adopted children in their sample, which are not representative of other children. Adoption studies are difficult and time-consuming as well as expensive, and there aren't many adopted children. It's not reliable. Extraneous variables do affect the study. Confounding variables also affect the study. Environmental determinism, the idea that your upbringing shapes who you become, and only your upbringing. A mixture of environmental and genetic factors is a better explanation. Children cannot consent to be studied, but adoption studies usually proceed with presumptive consent from the adopted parents and, where possible, the biological parents too. Adoption studies may create a rift between families, drawing attention between the differences between the adopted children and their stepbrothers or sisters, or drawing unfortunate comparisons between the biological parents and the adopted parents. This can create self-doubt, which the BPS warns about. Adoption study. Edexcel. The aim was to see if there's a genetic basis for schizophrenia. Procedure. They used the Danish family's records to locate adoptive and biological relatives of all participants. They tracked down 163 relatives and assessed their mental status. Psychiatrists blindly assigned and diagnosed schizophrenic patients into adopted schizophrenia, not adopted schizophrenia, biological schizophrenia, and not biological schizophrenia. Results showed that schizophrenia seemed to have a genetic component, as adoptees were more likely to have schizophrenia in their biological family rather than their adoptive family. Strengths and weaknesses. Strengths. A large sample ranging from teens to women in their 40s. It was replicated in 1975, 1978, and 1944, and they all found similar results. Concurrent validity from previous studies and contrasting from theories. Ethics. The data was only analysed, and it benefits society to know that schizophrenia has a genetic basis. Weakness. We have a social responsibility not to cause harm with these studies. Also, the study is not generalisable due to the location. It was in Denmark, therefore the participants were mostly white. It has vague and unclear concepts. Schizophrenia is a spectrum. The diagnostic techniques were not scientific due to some participants fitting into multiple categories. Brendgen et al. 2005, examining genetic and environmental effects on social aggression. This is also your twin study. Social aggression is aggression that socially manipulates behaviour, which can be overtly or covertly done. There's not a lot of research done on social aggression. A lot is done on physical or verbal aggression. Brendgen aimed to see if social aggression was genetic, to see if social aggression was caused by the same way that physical aggression was, to see what type of aggression would lead to another. Participants and procedure. 322 pairs of twins were tested, but complete data was collected of 234 of these twins. 123 pairs of these twins were DNA tested, and this backed up the assignment of MZ and DZ twins 94% of this time. This was considered reliable enough. 44 MZ males and 50 MZ females, 41 MZ males and 32 MZ males, and 67 mixed. It was a longitudinal study at 5 months, 18 months, 48 months, and 60 months, and then again at 6 years old. The data gathered considered the ratings of the twins' behaviours from teachers and classmates at 6 years old in the spring term to ensure that they were well known. Teachers' ratings were on 2 scales, and children gave scores based on physical and social aggression. The preschool social behaviour scale, PSBST, was used, and the direct and indirect aggression scale was used. These tests included statements like, tries to make child dislike other children, says best things or spreads nasty rumours about other child, becomes friends with other children for revenge, gets into fights, physically attacks others, or hits and bites others. You can see how this is split into physical and social aggression. Pair ratings were done by giving children a booklet containing photos of every child in the class and asked them to circle 3 pictures of children that were matched on 4 behaviour descriptions. For example, tells child not to play with other children, is mean in secret, gets into fights. Evaluate this procedure and the participants. There is a 94% chance that they were in the right category. However, there's still a 6% chance that they weren't. It has good representation of twins. It's a longitudinal study. Some of them dropped out, some of them were from underprivileged backgrounds, and this is low generalizability and reduces individual differences. Both scales used were well established. Pair ratings. This can lead to bias, and also it has an ethical issue. Pair ratings may lead to bias as people may not like the other people in the study. Does that make sense? The peers who were rating the kids may not like them, therefore leading to lower ratings. It's not actually based on the aggression. Ethics. It also may damage friendships and lead to isolation of participants due to them seeing as a bad kid. There's a high correlation between MZ twins and physical aggression, then the ratings on DZ twins and physical aggression. Scores for social aggression were similar between MZ twins and DZ twins. Physical aggression can lead to social aggression, but not vice versa. Teacher ratings did rate boys more physically aggressive than girls, and girls more socially aggressive than boys. Pair ratings from classmates showed that boys were more physically and socially aggressive than girls. There is also a moderate correlation between the teacher and pair ratings for physical and social aggression. This shows that it can be reliable. We can conclude from this that social aggression is due to social norms. They did not choose the participants. They took it from the Quinn-Neubeck twin study, a cluster sample. This reduces bias and leads to validity. They used inter-rater reliability. 2 researchers visited the classroom. This led to less bias. They used already established questionnaires. This means it was a reliable test of social and physical aggression. Early intervention could be characterized by the information from these studies. Social aggression is linked to environment. Then it's possible to reduce the effects of social aggression. The study takes a more holistic approach, including environmental factors. The study uses 2 different sources to gain insight, teachers and children, and they had a moderate correlation, making it more reliable. Which translations may have impacted the data? The study was carried out in Quebec, where some schools speak French and some schools speak English. The questionnaires had to be translated. We don't know if the participants actually understood. This impacts validity. The age of participants made data collecting a long process. 6-year-olds had just learned how to read and write, and researchers had to sit with the children and explain each question and make sure that the data was recorded. Therefore, makes the study a very long and expensive process to partake in. Therefore, this makes the study very hard to replicate and non-falsifiable. This makes the study unreliable. Extraneous variables, children's life, are not accounted for and only looks at the nature. This makes it low in validity and reductionist. It only investigated one age group. Research suggests that physical aggression takes place at the age of 4 and that social aggression doesn't fully develop until the age of 8. There's not enough age groups to say that this is an accurate study of aggression. Even though there was a correlation does not prove causation. Cause and effect, biology or environment. Therefore, this study is low in validity. The study may have caused friendship issues. Choosing who's the meanest kid. Kids don't normally think about this. Therefore, this study does not protect participants from harm, as it may set up twins to have a harder life than they would have. Scanning. Now you need to know what scans are and the strengths and weaknesses of scanning. Scans, also known as position emission chromatography, injects a person with a small amount of radioactive material to conduct a scan. The radioactive substance is attached to the glucose. A task is given to patients to do, whilst the glucose gets used up and the radioactive substance breaks down and emits positions. Where there's a high concentration of gamma rays, there's a high concentration of activity. This is where glucose will be used up. This is shown by warmer colours on the PET scan. You can see this on Raine's actual PET scans. Strengths of using PET scans is that it can help predict what issues patients may face in relation to brain activity. They also detect areas of damage indicated by which parts of the brain show abnormality. However, a weakness of using PET scans is that you're not advised to have too many, as it's unclear the future effects. And it's an invasive technique, and you may interpret them incorrectly. Or, if you know the results of the study, you may interpret them biasly. Computerized axial tomography, or CAT scans, can be used to take images of any part of the body by passing X-rays through the part of the brain at multiple different angles. The information is then interpreted by a computer and detailed images of the structure of the brain is created. Strengths. It provides information needed for surgery. It gives an accurate detail of brain structure, and It removes the need for surgery, and it's quick. Weaknesses. Pregnant women can't use it. It should only be used when the benefit of diagnosis outweighs the risk, and it leads to exposure to radiation. Functional magnetic resonance imaging, fMRI. Brain imaging is associated with blood flow. The head is placed inside a large magnet. Inside the magnet, the field of the nuclei with hydrogen molecules align with the magnetic field. As initial activity increases in the brain's blood flow, increases in the area to keep up with the demand of O2. When hemoglobin carries around the O2, it repels the magnetic field. When deoxygenated, it extracts the magnetic field and changes are detected by the scan. Strengths. No risk and it's non-invasive. Weaknesses. Pregnant women or people with metal cannot get in MRIs, or claustrophobic people. Correlations. Now you need to know the types of correlations and the strengths and the weaknesses of them. Correlation is designed to look at a relationship between two variables. A covariable are two variables that can be plotted against each other to indicate what type of relationship is between them. Researchers looking for the relationship between covariables or the way between these covariables can change when examined together. Tell us about the nature of the relationship between them. Correlations are plotted on a scattergram and are measured using a coefficient. Coefficient values range from -1 to 1. A correlation of -1 is a perfect negative correlation, and a coefficient is a perfect negative correlation. Positive correlation. As one variable increases, so does the other. Negative correlation. As one variable increases, the other variable decreases. No correlation. There is no association between the two variables. Strengths and weaknesses of using correlations. Strengths. They may indicate a connection between two issues in a situation where experimental proof is impossible. High in ecological validity, these numbers came from real life. They do not require manipulation of variables, and they are therefore safer and more ethical than doing experiments on humans. Weaknesses. They don't show a causal relationship, and there's no manipulation of an IV. There's no cause and effect. Therefore, they could be down to other factors, and they may be a relationship we may not know about, and they may be a third variable. Therefore, it's not reliable. As said, they could be a third variable impacting results, and they do not reflect a causal relationship, so we cannot see different views. Key question for biological psychology. How can explanations of aggression be used to reduce violence? Whitman had a tumour suppressing their limbic system, which meant they went on a violent killing spree and killed 17 people. This shows how the brain is responsible for emotional response, increased violence, suggesting nature is the cause of aggression. Females should be less physically aggressive due to their evolutionary disadvantage due to their nature, whereas males tend to be more aggressive due to being stronger. In the UK, males make up approximately 96% of the prison population, whereas females represent 4%. SSRIs can be used to reduce aggression. They release serotonin, they stop verbal aggression and decrease anger. Carver and Miller 2006 found that serotonin deficiencies were found among antisocial, aggressive and impulsive people. This suggests low serotonin can lead to psychopathy. Psychodynamic. Sports intervention catharsis can reduce aggression of children and adolescents. The limbic system plays a role in self-preservation as well as fight and flight. Early intervention may lead to you being able to address behavioral issues and reduce the likelihood of violent behavior. Medication or behavior to inhibit or hinder a brain structure function to reduce aggression. Evolution and natural selection. Successful humans were those best suited for their environment, and therefore they had to be aggressive to reproduce. Therefore, aggressive behavior and traits were passed down. By understanding the underlying cause and drives of someone's aggression can therefore be directed into a less harmful source. Testosterone increases competitiveness. Moser and Booth, 1998, saw that males with high testosterone were more likely to be criminals. Serotonin. Low levels of serotonin can lead to aggression. The psychodynamic approach states that the I did/the unconscious is entirely driven by impulses, with no thought for consequences. If the I did is denied pleasure, it becomes frustrated, which can lead to aggression. Psychoanalysts help direct clients to explore their own dreams and childhood memories to work out what they mean. This can help treat aggression. Andy and Vitali stimulated parts of the limbic system, the hypothalamus and basal ganglia in cats. Animals had aggressive seizures. However, using animal evidence to speak about human behaviour is reductionist. However, they do share basic functioning, and case studies do coincide. Evolutionary support. Daly and Watson, 1988. Young wives are more likely to be killed than older ones. However, it cannot explain cultural differences. In Brazil, aggression is more accepted. Dabbs, et al., 1978. Measured the levels of testosterone in violent and non-violent crimes and found higher levels in violent crimes. Egger gave females a dose of testosterone. Testosterone did not cause aggression. This means this theory is limiting in explaining the cause of aggression. Psychodynamic can be used to treat aggression by looking into the unconscious. Therefore, it has application. However, catharsis doesn't explain aggression. Practical investigation. A correlation into age and the attitudes towards drug use. I know this is all I put down, however, you should know your own practical investigation. You should know the strengths and weaknesses of your practical investigation. You should know how to improve it. I cannot talk about what you did for your practical investigation. However, I can say that you all used a correlative study, you all used quantitative data, and you all analysed with Spearman Zero. Next are all the questions that have come up from 2017 to 2024, including the specimen in terms of what topic they come up in.